Hello. Alternative splicing does provide the ability to produce diverse proteins from one given gene, and that increases the repertoire of the ability to produce and perform different functions even with less amount of genetic information. But when should and how should the genetic information or the splicing should occur? So we have, if there are these many probabilities or possibilities of splicing to occur, that is normal, exon skipped or exon extended, and intron retained or alternative exons splicing, then which one should it happen? At what time? At which points of time should which splicing should occur? And in which cells should it occur that way? These are some of the questions that happen. There are two ways things happen. Some of the alternative splicing can occur constitutively, meaning that all the time. So one gene could produce a normal one as well as this one, the exon skipped one, for example, continuously. And those would have been called as constitutive alternative splicing. Whereas if there is a scenario where some of the cells, in some cells we would see that normal one is produced in some cells, whereas in some other cells we would see that the intron is retained. That means it's a spatial organization in the sense that in some type of cells you'll have, from the same gene though, you would have a normal splicing type, whereas in a different cell type, we may have the whole intron retained type. That means splicing between 1 and 2 exons is not happening. Thereby, the intron is retained. We'll see a few examples. We'll just focus on the mechanisms only. So the first one is about the constitutive splicing. Here we have for HSV antigen, where we have this HIV antigen, T antigen, and capital T and small t. Here is capital T and here we have the capital small t. So the genetic organization is something similar to that of, this is an example of constitutive for exon extended exon. Say here we have the start codon, and this is the exon 1, say, and this is exon 2. We would have assumed there is a stop codon here. So at this border we have the 5' splice site, and here we would have at the 3' splice site. There is also a splice site like sequence which is within the intron, and so the splicing could occur between the 5' splice site here and the 3' splice site. But it could also occur, splicing could also occur between the pseudo splice site and this one. So there are two alternate forms for splicing here. So if splicing did not occur, if splicing occurred between these two regions, that is, you would have had a splitter splice site here and the 3' splice site. This would have been the 5' and 3' splice sites. And if splicing occurred between these two, we would have retained or had this much of the intron retained. So we would call that as the whole exon extended type. So if you see this, one of the sequence points is that the intron also has a stop codon within it. Stop codon, we would say it because if the ribosome were reading the mRNA, say from here, it will constantly keep reading until any sequence, a sequence that is of stop codon comes by. And here there is a stop codon that is in frame with the exon 1. So because of which, when the translation ribosome comes up to that point, it will stop translation. Translation termination would occur. As a result, a smaller protein is produced, therefore it is called as small t Ag antigen. Whereas if splicing occurred between the actual 5' splice site and the 3' splice site, then we would have proper splicing between exon 1 and exon 2. This is a larger open reading frame compared to what we had in this case here. As a result, ribosome would start translating and it will only experience a stop codon at the end of the exon 2. Therefore, it produces a larger protein, which is capital T antigen. So these two occur at the same time and continuously. So you have two different proteins produced from one gene continuously. So we call it as constitutive. There is also another example for it is the troponin gene, the enzyme, the protein that is functional in our muscles. So in this case, we have this is the pre-mRNA. It is showing exon 1, 2, 3, 4, and 5. So we have 5 exons, and in one of the cases in beta troponin, or we'll just beta troponin, we have exon 1, 2, and we have 4 and 5. So this is type of exon skipped. So the whole exon is skipped from here. This occurs in a way, it's the 5' splice site here at the end of or exon 2, and splicing occurs between these two regions or splice sites, this and this one. Therefore, the whole exon 3 is also eliminated in this process. So we have a different type of mRNA which has a continuous or an open reading frame, and therefore it would produce a protein. Alternatively, things may also happen differently. Splicing may also happen differently. We have exon 1, 2 is present, and 3 is present, and we have 5. So here we have exon 4 is missing. This could occur if splicing had occurred between this 5' splice site at the end of exon 3 and the 3' splice site at the beginning of exon 5. So the whole exon 4 is skipped. So this is kind of mutual exclusive also. Either it will produce a mRNA with exon 1, 2, 3, and 5, or it will produce 1, 2, 4, and 5. So unlikely that you will see a protein, a mRNA having both 3 and 4 in the same mRNA. So the larger one and the alpha troponin T and the beta troponin T are different in their amino acid sequences, because one of it, beta protein, has the proteins amino acids corresponding to exon 4, and whereas alpha troponin has amino acid sequence corresponding to that of exon 3. So these are constitutive because when we say constitutive, both types of alternate forms of these mRNAs are formed, and subsequently their proteins as well, in a single cell and all the time. The other ones are called as regulated ways, that is in regulated alternative splicing. We will have, these are controlled by certain proteins, and those proteins are referred to as, or factors you can say, as activators, and there are activators, repressors, enhancers also, several of them. We'll quickly have a small, a little bit look at some of the general mechanisms on that. Say here we have a mRNA transcript, that's the primary transcript, RNA transcript. It has the 5' splice site, and it also has 3' splice site. But there is also one site here, very nearby, onto which a protein could bind, and that is, the protein is, of course, here it's called as the repressor. So the normally the splicing machinery would recognize the 5' splice site, 3' splice site, and then splicing between the exons would have occurred. If the problem here arises because of this region, in this region we have both the 3' splice site as well as the site for a repressor is also present in this nearby or adjacent location. So if one protein is bound, there is not enough space for the other one. That is what happens well, let's just go ahead with a normal one. Say for example, if there is no repressor, so splicing machinery can bind here and also here, and splicing would occur, and now we have, say for example, exon 1 and exon 2 spliced together. In the other case, in a different cell, maybe there was expression of repressor. If repressor is produced, and repressor would bind to the site that is present here. Because of that repressor binding to the site adjacent to 3' splice site in this case, the ability for the splicing machinery to bind to the 3' splice site or even recognize is inhibited. Therefore, there would not be any splicing in this point, and the mRNA could remain as such. So you can call this as intron retained one. So here it is regulated. It is regulated by a repressor, and we call it as a repressor because it is repressing the splicing happening and obstructing the splicing. So we call it as repressor. You can say it as repressor of splicing. There are also other types of proteins which can enhance the splicing to occur. This is especially important if we have a little bit when the splice sites, 5' splice site and 3' splice site are not very they have sequence disparities. So here we have exon 1 and exon 2, and there is a splicing site here. The problem is that the splicing, assume that the splicing, the sequence is not very well recognized. So the affinity for the splicing machinery to recognize this region would be very less. So splicing might not occur at all. And what we might have because of it is exon 1, exon 2, and this whole intron is retained. It could occur like that. And why is it occurring? Because of the inability of the splicing machinery to efficiently recognize the 3' splice site in this case. There is also what may happen, however, is there might be activators of splicing machinery. Activators bind to certain regions that we call them as enhancers, like the exon splicing enhancers, etc. These are present usually within the exons, and when they bind here, when there is production of activator in the cell, it would recognize the splicing enhancer, it would bind there, and the activator also has affinity for splicing machinery, so it will bring in the splicing proteins, ribonucleoproteins proteins here, and will allow the splicing to occur between these two. So the presence or absence of the activator is determining if splicing should occur between the exons, and that's why we call them as activator. If we quickly compare that of the activator and the repressor, if the presence of repressor would inhibit splicing to occur, which otherwise by default would have occurred. The activators, the splicing would by default would not have occurred, but because of their presence, they would make the splicing to occur. So why do we call them regulated is not all cells would be having the expression of either repressor or activator, as the case may be. In some cells, if there is expression of repressor, then we would end up with unspliced mRNA. And in some cases, as this one shows, if there is activator production, then we would have a spliced one like this. So it all depends on the context of the introns. We cannot generalize it and say that for all mRNAs this is the case. It depends on the organization of the introns, the sequences, the consensus sequences that are present for 5' splice site, 3' splice site, and also the organization. How close in proximity are these enhancers or repressor sites or enhancers are present. We'll see you again. Thank you very much.